Hi everybody, welcome to Genome Informatics. Well, sorry to welcome you a little bit late, but it gives me huge pleasure today to welcome our first keynote speaker at Genome Informatics 2025. That's Marinka Zitnik. Marinka is an associate professor of biomedical informatics at Harvard Medical School and the Broad Institute. She's won a string of awards, and if I were to say she works at the intersection of AI and biology, that would massively oversimplify and undersell her achievements and the incredible breadth of her work. I won't list all the topics. I think you're probably going to get a taste of that now. But I'll just say it's a great honor that she's here to speak to us today, and welcome her to the stage.
Hello, everyone. Thank you for this kind introduction. Really excited to be here with you. Let's see if we can set up the slides. Okay, as I said, excited to be here. Really a pleasure to hearing about this fantastic fellowship that allows rising star students to attend these important meetings and learn about new things. So today I will talk about the use of AI to accelerate and empower biomedical discovery and how we are approaching developing new models that can leverage the recent advances towards drug discovery, development, and understanding of the roles and functions of molecules in various contexts. I will highlight several projects throughout the seminar, and these all come with open data sets, open evaluation benchmarks, model checkpoints, often also API endpoints, so you can connect your agents to our models and vice versa, and you're encouraged to go to our lab website to learn more. So we have seen over the last several years in particular the integration of recent paradigms of AI in biomedical workflows, and these models are beginning to have an impact on AI in two different ways. One is that they can change the way we go about our everyday research activities by using some of these models to help us potentially perform rudimentary analysis or retrieve and search for literature, etc. And the second important way how AI is kind of playing a role in biomedical discovery is that it is augmenting research and providing insights that might not have been possible with traditional deep learning models just a few years ago. And I will highlight what that means in this talk with a few concrete examples. Broad vision in our group is to develop what we refer to AI scientists. This is also known in the broader field as AI co-pilots or AI co-scientists that would be compound AI systems that are capable of skeptical learning, that can integrate multimodal experimental data with literature, and work alongside human scientists as their collaborative partners. Eventually, those type of models would be able to perform certain analysis by themselves. And so this is a vision that biomedical AI is broadly excited about since mid last year. And so what I would like to share with you today are 4 examples of models that we have recently developed that paved the way towards realizing this vision. And we will start with models that operate at the level of molecules and can help us understand and reason the functions of proteins across the entire human protein space. Then we will go one level up and look at the role of molecular interactions, as we know that molecules don't independently act of each other, but they form complexes that then play important roles in biological processes. And then we will go one level up and see how those biological interactions take place and are embedded in specific microenvironments that are defined by cells and cell type context. And finally, I will conclude with models that can help us reason at the tissue and body level and match patients to optimal therapies and recommend new therapies that would be effective and safe for diseases that currently have very limited treatment options. So with that, let's start with the first part of the talk, which is that of molecular reasoning. So the motivation for this part is observation that as a research field, we now have AI models that are able to predict protein structure from amino acid sequence and achieve atom level accuracy that is matched with experimental accuracy, and can do so for many, not all, but many important classes of proteins and many important protein families. And so this grand challenge that we can think of as developing models that predict structure for every protein has been one of the poster childs of success of AI for biology over the last several years, and also awarded with Nobel Prize last year. There is another grand challenge, however, that is largely not tackled with the kind of foundation models that structure prediction has been addressed. And that is predicting the protein functions and understanding what is the role or phenotypes that proteins perform by considering sequence and structure, experimental or predicted. And so the question becomes, can we also develop models that are able to predict function for every protein? Why would we want to do that? 20% of human proteins still lack well-defined functions, and many well-annotated proteins. By well-annotated, I mean that if you go to canonical databases of functional annotations, they might have a number of functional annotations there, but we don't really understand how those functional annotations vary across diverse biological states and biological contexts. 95% of all life sciences publications focus only on 5,000 most well-studied human proteins. So you can imagine there is a very long tail of proteins for which we have really limited, sparse information. So the question becomes, and the challenge is, can we develop the models that have the ability to predict, characterize, and generate functions for a broader range of proteins, ideally in a more universal manner? So I would like to share with you a model of that kind. This is a model that we have developed over the last year and a half. It's called Procyon, and it's an open source model that comes with open weights, training data sets, evaluation data sets as well. How can you think about Procyon? So Procyon is a multi-model foundation model, and I will unpack what that means through the next few slides. But it is a model that can predict functions, characterize, and even generate phenotypic descriptions. And when we say functions, what we mean are functions that broadly cover 5 knowledge domains that relate to understanding of biological processes, molecular functions, several components in which proteins are located, understanding disease mechanisms that proteins play a role in, mechanisms of actions of drugs, protein substructural domains, and molecular interactions. So when Procyon is trained, human user can interact with Procyon in conversational manner. So there are 3 modes of interaction between human scientists and Procyon that are shown on this slide. We can see in panel A is an example where user would interact with the model by specifying in natural language their prompt, where they can describe a particular biological phenotype that is of interest to them. And then Procyon will go in its internal database and it will prioritize, retrieve, and score proteins based on that description of the input prompt and rank them and present to the user. The second mode of interaction is interactive exchange or question answering session between Procyon and human scientists, where the model, when scientists can ask various questions and the model will engage with those questions by answering yes, no, and providing evidence for its own reasoning. And you can see here that in the input prompt of the model, the user can interleave both textual information as well as structural and sequence information. So the model is naturally multimodal because it allows user to specify their query by combining text with sequence and structure. And the third mode of collaboration or interaction between Procyon and user scientists is a capability of the model to generate new text. So this is a generative as opposed to predictive capability, where in the input prompt, user can provide a condition or class of biological functions and the information about the protein that they are interested in. And then the model will, through autoregressive sampling, generate phenotypic descriptions. We refer to them as protein cards. You can think of them as captions for proteins. I'm sure that many of you have seen how generative AI models can now generate captions for images of cats and dogs. So we're not here interested in doing generating captions for images of cats and dogs, but instead generating these phenotypic descriptions for proteins based on their multimodal data representations. Okay, so this is what happens, and this is how you can interact with Procyon once it's trained. But how do we train and develop the model? There are two critical questions here. One is, what is the training data set? The second one is, well, given this training data set, what is the model architecture of, and how do we tune parameters of that model architecture? So to train the model, we have created and curated Procyon training data set that consists of 33 million data points, where each data point is a tuple, and the tuple consists of two elements. The first element is information about the protein, which can be protein ID that is then linked to its sequence and structure. And the second item in that tuple is textual description of one specific function that the protein would have. This instruction tuning dataset covers 5 knowledge domains that are shown here in these boxes: functions, diseases, therapeutics, protein domains, and interactions. In addition to instruction tuning dataset, which couples together the textual information of functions with the structural and sequence data of proteins, to train the model, we also incorporate 250 million protein sequences that are used to train the protein-based language model component in Procyon, 800,000 protein structures that combine experimentally derived structures from PDB and predicted structures from AlphaFold that are predicted with high confidence values determined by PLDTT values, and 50 million full-length papers for natural language understanding. One critical feature of Procyon is that it does not pretend that protein amino acid sequence is just yet another English word with just weird 4 letters or 20 letters that would represent amino acids. Instead, the Procyon represents different data modalities, text, sequence, and structures by having dedicated tokenization strategies so that can take the input data, transform it into tokens, which are these units of analysis for large language models to understand and operate with now. And then it can consider then both modality-specific information and model and consider structure faithfully as well as textual information faithfully. So, given this data set, how does the model architecture of Procyon look like? So you can see this on the slide. We can read this from left to right. So fundamentally, user can ask questions that require Procyon to retrieve some information from its database, or users can ask questions and prompts that require the model to generate some free text. So based on the input prompt, it's always that elements of the input prompt are decomposed into inputs that consist of different modalities. So if there is then amino sequence information provided in the input prompt, it would be parsed through by the protein language model, which is this blue box protein LM that is trained on the amino acid sequence, and the structural information would be passed through a structural encoder, which is an atom-level geometric deep learning model, GearNet in this case, and the text-based portion of input prompt is processed by a text-based large language model encoder, which is the yellow box here. The outputs of these different encoders are modality-specific embeddings or encodings, where we have an embedding of the text, of the structure, and the sequence, and they are jointly optimized so that we get a unified embedding space where the similarity between text and sequence or text and structure embeddings reflect the structure of the training data set. So in the case of protein retrieval tasks, the model would then retrieve and score from this unified text phenotype embedding space proteins and score them, present to the user. In the case the model has to operate in generative manner, meaning generate the text, then we rely on autoregressive sampling to conditionally generate the text based on the user prompt, and that produces phenotypic descriptions. So to train parameters of these different boxes that we have seen in the previous slide that are modules or components of the neural model, we follow 3 key objectives. The 3 key objectives are the following. So first objective in the overall loss function is the language modeling objective that is designed for the model to understand and be able to process natural language and then also write coherent sentences using scientific language. The second objective is contextual protein retrieval. This objective technically is a form of contrastive learning objective, which is a popular way to perform training of large models nowadays without having labeled information for each and every data point. So through the mechanism of contrasting proteins that likely share functions and pushing them further away, further close to each other in the embedding space, and those that do not share further away from the embedding space. In the embedding space, that's called contrastive learning, the model is optimized to perform well on a variety of retrieval tasks. The third objective in the overall loss function is instruction tuning. The purpose of this objective in modern large language models is to ensure that models follow human instruction. So when the humans ask it to perform and answer specific question, they would really answer that question as much as possible and not deviate from it. So given these three objectives and the instruction tuning data set, we have trained Procyon model for several months on over around 200 GPUs and then produced an 11 billion parameter model that we have made publicly available. So what can we do with this model that we could not do with much, much simpler models for protein function prediction? Let me show you a few examples, and before that, I want first to show a brief snippet of results of model benchmarking. There are many, many plots, 30 pages of plots like that you can find in the paper that we have generated in order to rigorously benchmark and evaluate performance of the model. So what you can see on this specific slide, in these 6 bar plots on the left part, we can see performance of Procyon measured as Fmax retrieval score, so higher value indicates better performance. And performance is measured in a way that we have a number of protein tasks or protein challenges, and then we ask how often the model for a given protein, how accurately it can predict protein functions, cellular components, biological pathways, disease mechanisms or disease associations for that protein, drug target mechanisms, and drug protein binding events. Now, there is lots of thought put into that. The major challenge is how to deal with potential information leakage if models are trained on everything on the internet or everything in scientific literature. So we have performed 2 types of experiments to account for that. First is that we are running time machine experiments where we know what is the knowledge cutoff date for the data that was used to train the model, and then the model is making predictions after that knowledge cutoff date, and then those predictions are being evaluated. And second is we have a number of analyses where we stratified performance of the model based on protein or functional similarity that then allow us to get a sense of how the model would generalize when it would be asked to predict in functions for proteins that does not share strong sequence or structural similarity with those proteins the model encountered in the training set. Very briefly, what this summary plot shows, we can see on y-axis different models. Procyon is at the bottom, shown in purple. The models above are state-of-the-art modality-specific models. So here we have state-of-the-art protein language models that are single modality based on protein language sequence information alone. In this case, ESM3 that are fine-tuned with either MLP, shallow transformers, or directly used in zero-shot manner to KNN scoring. Then we also have structure-based only models. So GearNet is an atomistic model that can predict functions based on structural properties of the molecules. And then we also compare performance to widely used strategies that use guilty by association principle plus BLAST to transfer functional annotations from one protein to another. One interesting example that I can show here of that time machine experiment that I mentioned. So here we took one concrete example, which was a STING protein and a novel function that STING plays has a role in the context of immune system that was published 6 months after the knowledge cutoff date for the model. So it was the kind of test that allow us to be more confident that there is no information leakage about the function of this particular function of STING in the model. And so what we have done is we have designed a series of our collaborators who have characterized this thing have designed a series of text prompts in which they described only the basic phenomenon of the biological function. And then we can see here in this plot in the middle how that textual embedding of the basic phenomenon is embedded as a yellow triangle quite far away from the embedding of the sting protein. But then increasingly, as one makes the text prompt increasingly more precise by adding mechanistic insights in addition to basic phenomenon description of this function, as well as providing more precise function. That description, that will slowly move the textual prompt closer to where the embedding of the protein is. And so this is the intuition that this model is realizing to then be able to retrieve very highly, meaning with a high percentile rank, as we can see on the left bar plots, sting as the one that would satisfy the prompt described in the input. The more precise detail the prompt is, the higher the retrieval score, which is exactly what we would want to achieve with the contrastive learning objective that I have shown. One benefit of a conversational approach for protein function prediction is unbound vocabulary of user input. Practically, that means that we are not limited a priori to predict functions that coincide or correspond to predefined gene ontology terms or predefined reactome pathways. And this is because the function can be described as in plain language format. So here our collaborators have looked at a couple of proteins. Here is an example shown for FOXC1 that have pleiotropic functions that do not correspond to any individual gene ontology term. And then we looked at how well Procyon performs in terms of percentile rank retrieval of this protein when it is presented in the input prompt with 2 functions, 3 functions, or 4 functions, and whether performance of that is better than if we would just separately predict the association between that protein with each function and its representative gene ontology term, and then aggregate those results through averaging or rank aggregation strategy. And we can see here lift in performance in purple relative to orange lines for this bars for this experiment. So beyond benchmarking and kind of systematic evaluation that I have illustrated in the last 2 slides, we have made the model publicly available, and over the last several months, a number of collaborators in the US and Europe have been using the model for their own interesting questions that they want to ask. So here are a few examples of those questions. So in this example, our collaborator was interested in characterizing the binding between small molecule drugs and specific structural domains of proteins. So they designed the user prompt, which is shown here in the left part of the slide, where they presented to the model the structural information about the small molecule drug, in this case miglitol. They plugged in a library of domains of a protein, MGAM, and asked Procyon to rank domains based on whether which one is predicted to be bound by miglitol. And here, indeed, the top-ranked domain of MGAM was the one that is bound by miglitol. We have replicated this experiment now for a number of multi-domain proteins and found that those rank 1 domains predicted by the model and did correspond to those that are bound by the drugs in the case of these proteins. In another example, we have our collaborators who are working in Parkinson's disease were interested in characterizing and understanding AKNAD1, which has been recently implicated in the onset and progression of Parkinson's disease. However, when we look at AKNAD1 in SwissProt and UniProt, it is poorly functionally characterized. So there is not much information for AKNAD1, and at the time it has low association score or a functional completeness score in those databases. So what we have done is the collaborators designed the user prompt, and then we plugged that prompt into Procyon and instruct Procyon to generate free-form text descriptions or these phenotypic cards for AKNAD1. And so shown on the top is one example about the role AKNAD1 plays with information of actin cytoskeleton, which we have then independently examined by looking at subcellular images on subcellular localization of this protein in Human Protein Atlas. But that now allows us to go to thousands of proteins that have empty functional pages in canonical databases and generate this type of protein cards. Finally, the last example I will show here is the work that we completed just a few weeks ago in collaboration with neuroscientists and neurologists from Imperial College London and King's College London. Those were Owen and Nicholas labs. And so these two labs are interested in characterizing targets for multiple sclerosis. And specifically, they are interested in identifying specific disease targets that are associated with different pathology in MS. And so as part of that, what they wrote down 5 different prompts. The beginning of those prompts are shown on the left part of the slide. Each prompt describes a distinct pathology in MS. And they use Procyon to score and retrieve proteins that Procyon would believe would be those targets for the 5 pathologies here. So how did they validate whether those top ranked proteins make sense in the context of specific pathologies? So they took 280 samples of post-mortem human brain of patients who have died from MS, and they stained them and then performed RNA-seq experiments on those samples, characterized them into which of those 5 pathology groups each sample belongs, and then characterized through RNA-seq those samples, and then we looked at whether those differentially expressed genes as identified in each pathology were indeed retrieved, correspond to those proteins that were predicted highly by Procyon. And indeed we can see that happens here. So this box plot that is shown in the middle of the slide shows what is the percentile retrieval of proteins that were validated to this RNA-seq experiment in Procyon, where the Procyon predictions were done of course, before the post-mortem analysis, staining, and RNA-seq was completed. And so beyond that, they have then used Procyon in generative manner to help generate protein cards for each of those highly ranked proteins that can help them reason about the role of proteins in these specific pathologies. So just to summarize, Procyon is a general purpose, or goes towards a general purpose model for predicting protein phenotypes, generating and retrieving protein phenotypes. There are a number of applications beyond a handful that I have shown to you that we have engaged with, working with our collaborators across different diseases and protein family classes, where they have used Procyon to perform protein function retrieval, generate phenotypic descriptions, look at the subcellular localizations of phenotypes, study pleiotropic functions of proteins, and as well as model and characterize proteins in the context of diseases or even certain drug mechanisms. And it's an open weight model, so everyone can build on it or build their own better model using the training data sets that we have released for the community. The limitation of Procyon, among various other things, is that the unit of analysis for Procyon is always one protein, and this is a human protein model. We would love to extend it to other species as well, and there are various challenges of how to do that effectively. But it can only reason about individual molecules and individual proteins. And we know that proteins don't act independently of each other, but they form molecular interactions. So how can we go one scale up and understand how molecules interact and what can tell us about the functions and roles that they are part of. And so here I would like to highlight the work that was recently completed by a stellar PhD student, Ada Feng, who's currently at DeepMind, and I hope she returns to the lab in a few months. So she developed Atomica model. So Atomica model is an atom scale model. So this is a model that operates at the level of atoms, and it's a model of molecular interactions. Okay, what does that mean? Atomica is specifically focused on looking at interaction interface between two molecules. So if we have an interaction complex here, we can look at the interaction interface by defining an interface of specific size. And then what we can do then is represent each interface by a graph. And so this graph has nodes that represents atoms in molecule 1, shown in blue, nodes that represent atoms in molecule 2, shown in yellow, and then edges between those atoms indicate physical constraints that represents bond with additional metadata, such as bond lengths or dihedral angles. We have a second level of nodes, which are higher order structures that represents blocks, such as common chemical motifs, or amino acids, or nucleotides. So the data set for Atomica to be trained on is then a data set of 2 million interaction complexes, where each complex is represented by this bi-level atom graph. So the data set is a data set of 2 million graphs that look like the one I shown in this example. Atomica is a geometric deep learning model because we have these graphs, and graphs represent physical systems and physical structures. We need to train an equivariant graph neural network, or here in this case, graph transformer model. And we train this model through self-supervised learning. So it's effectively, think of it as next token prediction as used for natural language, but we're not doing that on data that looks like a sequence of words, but we are doing that on a data set that looks like these structures. So practically that means that during the process of training, we take this graph-based representation of interaction complex, and we randomly mask the identity of one atom in that complex, and the model has to predict the identity of that atom or of that block. And we can do the same for edge properties as well. So technically, this objective is called one-step discrete diffusion objective, but it's a form of self-supervised objective that the model learns how to use the local and global information in the graph in order to predict what has been masked out. The key feature of Atomica that makes it stand apart from other geometric deep learning models for molecules nowadays is that the Atomica is trained on interaction complexes that go beyond protein-protein interactions. It is trained on complexes involving proteins, ions, lipids, small molecules, as well as nucleic acids. How is this possible? This is possible and is justified because the data are represented at the atom level. And so the atomistic representation of these different types of molecules allow us to train a single model for molecules of different sizes and for different molecular modalities. Once we pre-train the model, what do we get? We get this latent space that is a latent space of interaction complexes with 2 million points. Every point is represents an embedding of an interface of a pair of molecules. One thing to mention, and that we wanted to test specifically, is whether this type of multimodality training in Atomica model is useful. And so specifically what we found is that for modalities for which data are least abundant, such as nucleic acids, DNA and RNA, for which structure-based data on interactions with other molecules is least abundant in current databases, whether having the model that is trained on other non-nucleic acid modalities at the atom level can lift model performance for these data modalities for which data are sparse. So we designed an experiment where we have trained Atomica model several times, but each time we train it either on data from just one molecular modality, and then compare that to training the model on the entire data set. And what we can see here is strictly lifting model performance in being able to capture, in this case, the identity of masked blocks more accurately by having a model being trained on data from other modalities and effectively transfer that information to the target modality. What's most exciting here is that the strongest lift in performance, so this is this delta AUPRC, higher on y-axis, the stronger is improvement in performance. And we see the strongest improvement in performance for those modalities where the amount of data in the training data set is least abundant. And so that is interesting, exciting, and often explored in the context of AI within the broader vision of understanding scaling laws. And specifically, is there hope and potential benefit to training large models by putting lots of resources in training these large models? And what would that benefit be? And so here is an example of that in the context of molecules. After we evaluated and trained the model and performed numerous sanity checks and quality on the data sets and model outputs, we wanted to use the model to do something new and exciting. So here, Ada has used Atomica to scan through the dark human proteome and identified proteins that are most likely heme binders, as predicted by Atomica. And so Atomica predicted 5 of those binders, and then our collaborators in the chemistry department, Pentoliti Lab, were able to synthesize those proteins and then performed functional assay to test for whether they are heme binders. And so indeed, all 5 were verified as being heme binders. And what is most interesting here as well is that 4 out of 5 of those heme binders could not be identified using traditional approach of scanning the structure for structural motifs that are indicative of heme binding. And so this is one example of how then these models can have the potential to move beyond what a traditional standard approach, in this case scanning for the presence of absence of structural motif that is indicative of heme binding, would be able to achieve. Okay. So I have just 2 minutes, I suppose. So I will speak a bit about this very last dimension on therapeutic modeling and how we can really take the lessons learned from understanding the functions of individual molecules and characterizing molecular interactions and go all the way to human. And so our paradigm to do that nowadays is through the use of large language models that not only can analyze and fuse different types of data, but also can take actions in the world and can use different kinds of tools. And we often call those models agents. So how do we build those models? And is it challenging to build them? Yes, it's quite challenging to build them. So what we have just released is an ecosystem, an open ecosystem that would allow anyone, including those you here, to build your own agent model that can use tools, which we would call AI scientists, and connect that model with a broad range of scientific tools, over 600 tools that we have. So if you go to aiscientist.tools, you will see Tool Universe that is an ecosystem. It's in a protocol that allows you to connect your favorite AI model, large language model, reasoning model, open or closed form, with Tool Universe, which is a collection of 600 tools. Those tools are other machine learning models like Procyon or AlphaFold, or bioinformatics workflows like ScanSpy or Asiverse, or scientific packages, or visualization tools, or tools that can do literature search. And then this a large language model can then use tools in order to help with scientific tasks. Advantage of Tool Universe is that it can be connected with any large language model, whether it's open or closed form. So if you're a fan of Claude, you can connect it with Claude, you can connect it with Gemini, open source models like QN, or GPT or ChatGPT Codex. How does it actually work? So once you connect LLM of your choice with Tool Universe, what you get, if you connect it with Claude, you would kind of see something like this, where you see the usual prompt that you might be using every day, but there is an option below that says Tool Universe, and that is the 600 scientific tools that the model can now use, so that it will not hallucinate by just doing reasoning in literature only, but it will also be able to analyze data and retrieve information from curated resources in real time. What is the magic source? Or the critical component in innovation here? The critical component is a protocol that specifies how different AI models can communicate with each other. So in similar way as 20 plus years ago, the introduction of HTTP as a protocol has enabled the internet as we know it today by being able to go from static websites that were disconnected from each other to an ecosystem where websites can link to each other, and then you can get more complex platforms together. Now there is a time where we have AI tool interaction protocols, and these are protocols that the LLM uses in order to identify which tool is relevant for the task at hand, then go and execute that tool, then get results from that tool and interpret the results from that tool appropriately to perform next step of analysis. So I know that I'm late, so I will perhaps stop here. You can, if you go to AI Scientist.tools, you can try it out. And like the way just practically it looks like, I was just in the back the other hour, so I just kind of quickly built one. So here is one example of how GPT-5 is connected with a set of medical knowledge graphs that we have built that capture biomedical knowledge, including Prime KG, Optimus KG, and African KG for African-related healthcare and medical research. And then when one would ask a model about the reason for example, how variations in BDNF gene would impact the response of patients that are put on lithium, then the model would not only reason by reading literature and text and what it can find in internet or PDF or papers so that we write, but it would also use tools, and here in this case, look into the knowledge graphs that we have, and it would retrieve information from those knowledge graphs that come from curated human databases, and it would retrieve also information about the drugs that are being mentioned by user in the prompt, and then it will summarize and then provide the final response. This is just an example of what can be done by connecting, in this case, GPT-5 with a set of tools that corresponds to curated knowledge graphs. You could also connect it with your genomic knowledge graphs, for example. Okay, so let me go back here. I will conclude this talk by acknowledging a fantastic group of students that I'm fortunate to be able to work with, PhD students and postdocs in the group, and also our funders. And we have lots of open source tools that you are welcome to check out. Thank you.
Thank you so much. We do have time for questions. I see one right there.
Very interesting and exciting talk about the protein phenotype model. As I understand, it's using the already known information to predict the unknown information. And what I'm interested in is that can you predict percentage-wise for the total potential information, the percentage of known versus unknown is how much?
Yes, so it's a fantastic question. It's at the very core of lots of the discussions that are currently taking place in community, with AI scientists being also a hot topic for various commercial large language model providers, with the holy grail being arguing whether AI scientists generates novel hypotheses and what is really novel in scientific discovery. And it's incredibly hard and challenging to assess whether a specific hypothesis is really novel. So for example, a few weeks ago, it has been announced that GPT-5 Pro has solved mathematical problems that the Berkeley list of open mathematical problems is specifying as lists of open problems that humans have not solved yet. And that 2 or 3 problems on that list have been solved by GPT-5 Pro. So only a few days later, what we have realized is that what GPT-5 Pro has done, it was able to really effectively do literature search and found that those problems were actually solved by humans 20 years ago. But one of the problems was solved and described in a relatively small journal in mathematics in Hungary. And it seems like those who that nobody was aware of the solution for that problem to exist. So there's lots of speculation about what is novel scientific discovery, what is not, and what these models are currently capable of. I would say that we are potentially at the stage where these models can generate hypotheses that I would refer to them as connecting the dots type of hypothesis, where we might have disparate data resources or disparate areas of biological research or communities that do not necessarily talk to each other or live within the same namespace or live within the same database, and then models that are able to do accurate retrieval and hallucinate less and less by being grounded in prior knowledge can aggregate, summarize, distill that information and effectively like generate new hypothesis that is still grounded in prior knowledge and evidence of it, but it's somewhat new in the sense that the dots that were connected and brought together are novel.
How do you think about the potential for these sorts of models to be used by bad actors for, say, weapons development?
Yeah, so potential is high. So and it's challenging because these models are now increasingly available to broader audiences, and it's often our goal and ambition to make them available as much as possible. So you have seen the one that I showed at the end of the talk. If you go to chat.grants.ai, you can try it yourself right now. And you don't need to log in, you don't need to write code, you don't need to have access to GPUs. So just increasing decreasing the barriers for research, it democratizes access to these type of tools. It also creates opportunities for bad actors, which is to some extent true for many different technologies. I think it's important to explore opportunities for dual use. For example, before we released Tool Universe, we performed a series of safety evaluations through a technique known as red teaming, which is effectively that before we release the model and the system, we collaborated with a group who was not adversarial group, but was pretending to be adversarial group, and then they helped us evaluate what are the risks and specifically what are the tools that are in this database of 600 tools that would have higher potential of being used in adversarial manner. And of course, one would need to do that in a continual manner, which becomes very challenging. So it's a challenging balance to draw because we want to make these models broadly available on one end, but by making them so broadly available, it also means that you don't need necessarily a PhD degree now to optimize a molecule such that would be optimized with respect to being a potent bioweapon, for example. And just final point, of course, after we optimize the molecule to generative AI, going from that to actually synthesis of the molecule is a huge step. So that's still our guard, yes.
Thank you for the presentation. My question is whether your model predict numeric phenotypes instead of like some descriptive phenotypes?
Yeah, that's a fantastic question. Yeah, so we have Procyon, no, but we have now another model that I was not able to highlight that is called Prism that can predict numeric phenotypes. And by numeric phenotypes, what I mean are cellular phenotypes that are defined based on cell states or cellular descriptions, such as, for example, cell type abundance profiles, where we can, from a sample taken from a patient, we can characterize which cell types are present and what proportion of cells of specific state or types exist in sample, and then that is then the phenotype for the patient, for the Prism model, instead of being free-form text description of biological function. Another model that we have is called Compass, where phenotypes are given by transcriptional profiles of microenvironments of tumors for patients. And the model reasons about whether an individual with this type of tumor microenvironment, characterized by transcriptional profile of their tumor state, plus information about the patient, such as sex and age and medical history, how would they be likely responder to a specific type of immunotherapy?
Okay, maybe up there.
Thanks for a great talk. how is this technically built, your tool universe? Is it using an MCP server or something on top? And then the second question is, 600 are a lot already. How did you and who did curate the 600 tools, and how did you actually validate them? I saw you have like validation tags on the tools. Thank you.
Great question. Yes. So the current most well-known protocols for connecting different AI models to each other are two. One is called MCP, that was developed by Anthropic, and the other is called A2A, or agent-to-agent communication, that was developed by Google. So the AI-tool interaction in tool universe is distinct from those, and it's distinct in a ways because scientific tools present some unique challenges. Here's an example what I mean by that. What we have in science, we often have a number of competing tools with overlapping functionalities. So imagine that you want to use single-cell foundation models. And there are many. There is SSGPT, there is Transcript Former, there is Variant Former released a few days ago, UC, ANSI Foundation, etc. So which tool to select in a specific context? You might want to have the model the ability to select the tool that would potentially be trained on cell types that the question in the task that the user specified will be relevant for. And so this type of handling situations where we have many tools with partially overlapping functionality for the same task is not handled by MCP or by agent-to-agent protocols, because in the context of those LLM providers, they mainly focus just on two tools. One is writing code, and the second is doing deep research and crawling the internet. But with science, we have other type of problems. Another problem is that many tools that we have in science are stochastic. The output is not deterministic, which practically means that a tool that might have been proven useful so far, in the next question might not be useful at all. So it's not that the same tool has the same utility throughout the entire universe. And so these are the features that we are addressing with Tool Universe. So we have these individual components that we call tool finder, manager, caller, composer, to address the specific properties of scientific tools. We have 600 tools. We have validated that the model uses the tools correctly. We are not validating the tools themselves. So if the tool is AlphaFold, then we rely on the AlphaFold and the benchmarking they have done in that paper in our tool. But what we validate and ensure is that the models that would use the tool would use it correctly, meaning they would know what are the input parameters, be able to select them, and know what the output parameters are.
Okay, there's certainly more questions, but I think for the sake of time, what I'm going to suggest is Dr. Zitnik is going to be here for the rest of the day today.
Yes.
So you will have other chances to ask your questions. So please thank our keynote speaker again.